Good morning, dear students. Today we are starting a new module and our topic is fermentation and types of fermentation. Fermentation, it is a metabolic process that is taking place in microorganisms, and during this process they are converting the carbohydrate present in a suitable substrate into compounds like alcohol, acid, etc. And this process is mainly carried out by heterotrophic microorganisms like yeast, bacteria, etc. under anaerobic condition. And the science of fermentation, it is otherwise known as zymology or submerged. And Louis Pasteur is the French scientist who have discovered this zymology, and in 1856 he noted the role of yeast in fermentation process. So fermentation, it is a anaerobic process that is being carried out by microorganism, whereby they are converting carbohydrate substrates into carbohydrate rich food materials into alcohol, acids, enzymes, antibiotics, etc., which are of human use. And so many microorganisms are involved, mainly bacteria and fungi are involved in this fermentation process. And the science of fermentation, it is called zymology, and Louis Pasteur is the founder of zymology. That means that doesn't mean that zymology doesn't exist before Louis Pasteur. Louis Pasteur noted zymology for the first time, and he found out that yeast is responsible for fermenting so many foods, especially the wines. Then microorganisms in fermentation. Bacteria, they are able to convert carbohydrate into lactic acid. Yeast, they can produce so many alcohols like beer, wine, and other spirit. And fermentation, that process, it occurs naturally, and before human evolution this process is there in nature. But nowadays the fermentation has become a controlled process by human intervention. Types of fermentation. Mainly two types of fermentation are there: submerged fermentation and solid state or solid substrate fermentation. First one is submerged fermentation. Submerged fermentation, it is otherwise known as liquid state fermentation. It is called a liquid state fermentation because the whole process is taking place in a liquid medium, and it has been performed in larger tanks that are called bioreactors or fermenter. And this tank or fermenter is having a size that is ranging from 10,000 to 26,000 square feet. And this liquid state fermentation or submerged fermentation, it is carried out in industrial scale. And here this microorganism, it is being grown in the liquid medium, and the nutrients that is essential for the growth of microorganism, it is either provided as dissolved state, or it is provided as suspended particulate solids in the liquid medium. And peculiarity of this liquid media is that it is ideal for the culturing of unicellular organisms like bacteria and yeast. And since it is a liquid medium, constant supply of oxygen is required for the growth of microorganisms. So submerged fermentation is otherwise called liquid state fermentation because the growth of microorganism is taking place in a liquid media, and the substrate or the nutrients that are needed for the microbial growth is being supplied either as in the dissolved form or as particulate solids in this liquid media itself. And since it is a liquid media, the oxygen has to be supplied for the growth of microorganism. And this submerged fermentation, it is been taking place at industrial level, and so large bioreactors or fermenters are needed for the submerged fermentation to occur. Then submerged fermentation, here microorganisms are grown in liquid media like a free-flowing liquid substrate like corn, steep liquor, syrup, or nutrient broth. Nutrient broth means just like nutrient agar, it contains all the nutrients in a desired level for the growth of microorganism. Difference between the nutrient agar and the nutrient broth is that in nutrient agar, solidifying agent is being added so to convert it into a solid media. But in nutrient broth, since agar is absent, it will remain itself as a liquid media. So in submerged fermentation, this liquid broth or constip liquor or syrup are used as liquid media, and it is used for the production of the submerged fermentation. It is being conducted for the production of industrially needed enzymes, then antibiotics and other bioactive compounds. And microorganisms like bacteria which need much amount of moisture or wetness, they are suitable for the submerged fermentation. And an example of submerged fermentation is the commercial production of citric acid. Submerged fermentation process, it is been carried out in a closed container that is filled with this nutrient liquid broth, and it is inoculated means it is been mixed with suitable microorganisms such as bacteria or fungi, and oxygen is been supplied by stirring the fermentation media. And this microorganism, they will grow in the nutrients, and they when they grow, they will break down the nutrients in this broth into simpler compounds, and secondary metabolites will be produced, and it has been secreted into this broth by this microorganism. And the common substrate used in submerged fermentation are the soluble sugars, molasses, liquid media like fruit and vegetable juices. So these are the common substrate that we are used for fermentation. So in fermentation there are some terms like raw materials or substrate. That means the organic carbohydrate source that we are added for the microorganism to grow, and microorganism, they utilize this substrate for their growth, and they are converting this substrate into secondary metabolite that are useful to human beings. So substrate means their growth medium. Okay, so this commonly used substrate are sugars, that is soluble sugars, then molasses means കരിമ്പിൻ ചണ്ടി, then liquid media like fruit and vegetable juices. Types of submerged fermentation. Submerged fermentation is basically of two types: batch fermentation and continuous fermentation. Batch fermentation is again subdivided into fed batch fermentation. Batch fermentation is a closed system where the substrate and microorganism, they are added to a bioreactor at the initial stage, and they are not removed until the fermentation is complete. That means batch fermentation, the fermenter is there, and we are adding this microorganism and the substrate for the growth of the microorganism into the fermenter at the initial stage, and then this entire vessel is closed, and for a fixed time we are not disturbing the system, and the products or the materials that we need as the byproduct, they will be extracted or they are removed from the fermentation tank only when the whole fermentation process get completed. So that type of fermentation is called batch fermentation. In batch fermentation, microorganisms are inoculated to a fixed volume of medium in a fermenter. That means if we are taking, for example, if we are taking 500 ml of substrate in a fermenter, then 100 ml of the bacterial culture or microbial culture is being added to the 500 ml. That means if you are taking 1000 ml, we have to add 200 ml of microbial culture. So there is a fixed volume, a fixed ratio is maintained between the substrate we added as well as the microbial culture we inoculate. And as the microorganism grow, they will utilize these nutrients and they will produce byproducts, and the byproducts get accumulated, and they will mix with the broth, that is the liquid media there, and at the end of the process we will take the broth and purify the byproducts. So that is the peculiarity of batch fermentation. So the thing that you people have to remember is that in batch fermentation, it is a closed system where we are applying this inoculum, as well as inoculum means the microorganism, inoculum as well as the substrate at the initial phase. Then the entire vessel get closed until the entire fermentation process get completed, and then we open this fermentation tank, and the liquid content in this fermentation tank is being purified to extract the product. Then advantages of batch fermentation. Here substrate and microorganism are loaded into the fermentation batch wise. That means we are if we want to produce, for example, grape juice, wine, then we are putting grapes as the substrate, then yeast, Saccharomyces cerevisiae as the inoculum or the microbes, and then we are maintaining a fixed volume. That means for 1 kilogram of grapes we have to add 2 ounce of yeast, like that. Then the entire vessel is been closed for say 45 days, and then after that we are opening this vessel, and then we have to purify the wine. We have to extract the wine from the other waste materials that are present along with the wine. So that is an example of a batch fermentation. And here the substrate and the microbe is added at the initial phase only. Then we didn't disturb that system. Okay. Substrate and microorganisms are loaded into the fermentation batch wise. That means if you want to prepare wine second time, we have to clean the vessel, and we have to add new grapes and new yeast to that vessel. So batch wise we are adding this substrate as well as the microbe into a fermenter. Then other advantage is that low investment cost. The cost of production is very low when compared to other submerged fermentation. Then it is simple to control and operate. We can easily control and operate the entire fermentation process, and it is easy to maintain, and this process ensures a complete sterilization, because we are not handling the fermentation tank or the fermenter in between. So there is no chance for getting contamination. So risk of contamination can be avoided. So complete sterilization can be ensured. And this is the most popular method for the production of ethanol. So we can prepare ethanol in large scale by means of this batch fermentation. Disadvantages of batch fermentation: comparatively low cell density, because we are giving a fixed amount of substrate and fixed amount of inoculum. So only a fixed amount of nutrients is available. So what happens is that even though the bacteria want to divide, they won't able to proceed further, because only fixed amount of nutrient is there. So low cell density will be there, and comparatively long time gap between batches. So because we have to clean the entire vessel, we have to set up it again once more, and we have to sterilize the whole thing. So because these are all time taking process, so it is a very time lag. There should be a very long time gap between two batches. So these were the disadvantages. So low cell density and a long time gap between two fermentation process. Fed batch fermentation. Fed batch fermentation, it is a modified version of batch fermentation. Here microorganisms are inoculated and grown in nutrients for certain period, as in batch fermentation. So similar to batch culture, we are initially growing the microorganism in a substrate in a fermenter, in a closed fermenter for a certain period of time. So similar to that of batch fermentation. But then after having a fixed growth attained by the microorganism, we continuously add sterilized nutrients into this culture, microbial culture, throughout the remaining duration of the fermentation. So At the initial stage, we are just keeping microorganism and the substrate into the fermenter, and for a fixed period of time we would not interfere with their growth. Then after that, we on a regular basis we are adding the sterilized nutrients into the culture microbial or into the fermenter till the end of the fermentation. So here the cell density in the bioreactor will increase, and the rate of growth in the culture is maintained by adding nutrients. So it will reduce the risk of overflow metabolism. So in overflow metabolism, microbes will not able to utilize the substrate properly. But here in this fed batch fermentation, since the nutrients are being continuously added, this microorganism can avoid this overflow metabolism. They can continuously utilize the nutrients, and they can continuously divide into populations. Okay. So the fed batch fermentation is much more advantageous than batch fermentation. The difference is that here after a fixed period of time we are adding nutrients always into the medium to promote the growth of microorganisms. Peculiarities of fed batch fermentation. It is the most common mode of fermentation in bioprocess industry, and it is useful when we have to obtain high product yield, and it is preferred when the product is positively correlated with the microbial growth. If the microbial growth is high and then the product accumulation it will be high, then we can go for this fed batch fermentation. So if the product is positively correlated with the microbial growth rate, then fed batch is the most preferred type of fermentation process. And as the substrate is not overfed during the process, byproduct accumulation is limited. So we are not adding a larger quantity of substrate. We are adding substrate at regular intervals in fixed amount. So the overfeeding of this byproduct is being limited. So these are the peculiarities of this fed batch fermentation. This is the diagrammatic representation of submerged fermentation. Here the central cylinder shown is the fermenter, and we are introducing the media here. At first we have to prepare the media, then we have to sterilize the media, and the media is the substrate that is required for the microbial growth. It is being fed up into this fermenter, and then the microorganism that is growing in this media and converting this media into useful product is taken as inoculum, and we have to prepare the inoculum, and then we have to maintain the correct proportion of inoculum to be added along with the media. So the volume ratio has to be fixed there, and this inoculum is also being introduced into the fermentation tank, and there is a bioprocess control for checking the temperature, pH, and other factors inside the fermenter. Then after the fermentation process, so in submerged fermentation we know that it is taking place in a liquid medium. So the inoculum is spread throughout the medium, and in order to get the inoculum spread throughout the medium, we are using a stirrer. That is a fan shaped structure that is seen at the central portion. So it will continuously rotate to provide sufficient amount of oxygen to this microorganism. And then after the entire fermentation process has been carried out, we can recover the cell from the cell recovery unit, and we can also recover the product from the product recovery unit, and then the product is being purified and waste are removed. So this is the submerged fermentation, and it is this diagrammatic representation here is same for the batch fermentation also. And in fed batch fermentation, the difference is that here we are in batch fermentation, we are just adding the media and the inoculum only at the initial stage, and then the entire setup is in a closed condition. But in fed batch, we in between time intervals, we are adding this media. And we have to prepare the media, and we have to sterilize the media, and this media is added into this fermentation tank at regular intervals. So that is the difference in diagram for the fed batch fermentation. So in next class we will continue with this continuous fermentation. So Thank you.